Good morning, everybody. I think we are going to start this session today about presenting this month's tools of computational biology in CSIC. Okay, so I think it's time to start. So first, thanks for being here. And thank you to the organizers for this opportunity. So my name is Juan Fdez Recio, and I'm leading the group of Structural Informatics, Modeling and Biological Mechanisms at the Instituto de Ciencias de la Vid y del Vino, which is a mixed center, CSIC, Gobierno de La Rioja, and Universidad de La Rioja. And today I'm going to tell you about pyDock and other related tools that we have developed during these along the years for structural and energetics modeling of protein-protein interactions. And so I divided my talk in these three parts. So we start talking about the tools for structural modeling of protein interactions, and this is actually the focus of our research. So we aim to study protein-protein interactions, which are essential in all biological processes and also very important to understand the pathological situations at the molecular level. So we are developing and applying computational biology tools from different perspectives and integrating different disciplines like structural biology, biophysics, or molecular biology. And the main motivation for our research is the very limited structural coverage of protein-protein interactions. So if we compare with the human proteome, in which most of the proteins has a structure or a reasonable model that can be produced. So in the case of the protein-protein interactions that all these proteins are forming or potentially forming, the situation is very different, no? Because we have the structure for very few of all the potential protein-protein interactions that can be estimated to be formed in human and also in other organisms. So we can apply computational biology tools like homology-based modeling, but again, there is a strong limitation by the low number of templates that we can use for predictions. So we have to rely mostly on affinition modeling. And among all the possibilities that we have for affinition modeling, we disregard molecular dynamics because it's very costly and not very efficient for this type of predictions. So the community has focused on the development of protein-protein docking algorithms. So basically what we aim is to try to predict the structure of a complex formed between two proteins, starting from the unbound structures or reasonable models of the unbound proteins. And we have like a vague description of this process at the biological level, but we think that we can apply some strategies, some approximations, like considering, for instance, in a first phase, we can consider the proteins as rigid body. So we may think that at this stage, when the proteins are starting to interact, the internal conformation is not very relevant. So in that case, we can estimate the energy by by long-range interactions, electrostatics, or medium-range like dissolution. And then later, we can do more complex energy calculations, or even including some kind of flexibility. So this is, in general terms, our approach. And our methodology is pyDock. This is the methodology that we have been developing from many years ago and has been growing. So basically, what we do is to do some sampling of the rotational and orientational space between the two molecules using fast Fourier transform approaches that are external methods like pyDock, techDock, and there are many available. So in this way, we generate thousands of these potential orientations very fast, very quickly. And then we use a scoring function in order to try to identify the correct orientations. And this scoring function is based on energy. It's an approximated energy, but we don't want to to leave the energy considerations out. So we want to understand the process in physical terms. Basically, there are three terms: Lennard-Jones, a contribution from Lennard-Jones potential. This is van der Waals. Coulombic electrostatics, and dissolution, which is key in our success. No, but actually it's very simple because it's based. So it's a function of the atomic accessibilities of each atom and the atom type. So well, depending on the atom type, the contribution to the dissolution can be favorable or not. Okay, so usually apolar atoms are favorable contribution to dissolution, but polar atoms are disfavorable. So we defined different atomic solvation parameters for 10 atom types, and then when with this simple scheme and the parameters that were optimized almost 20 years ago, we are still using this basic approach for scoring. And is giving very competitive modeling. Okay, so this approach, pyDock, has been used through collaboration with different experimental groups to different systems of interest, biomedical and biotechnological interest, and has able to help to model some interactions between protein as well as protein with other biomacromolecules. But what we know is that in many cases this is working quite well, especially in those cases in which proteins are fairly rigid. Okay, if they interact as rigid bodies, then our approach is it's quite successful. But as long as when the proteins start to be more flexible, these predictive rates are dropping a lot. So we know that this is a strong limitation in our tools, in our pyDock, and also in many different docking approaches. But so even if predicting the binding mode in sometimes is difficult, what we found very interesting is that when we analyze the results of docking, all the possible models that we can generate, for instance, here we can visualize all these models. Every dot will be the center of coordinates of the ligand in all the different docking positions. We can have thousands of these models. And if we color according to energy, we see here in red the lowest energy docking models, which accumulate around the real position of the ligand. Okay, so this is we have observed this in many cases, and we are using this for interface prediction. Okay, so we are using this to identify the residues that are more often found at the lowest energy docking models. And this is giving us some number for each residue that very often corresponds with the real binding interfaces. Okay, so this we call this a normalized interface propensity, and is one of the modules that are more successful within the pyDock pipeline. We have many other functionalities and modules that are integrated in this pyDock pipeline. So in addition to the standard, the basic protein docking, we have built like a pipeline, no? A very modular setup in which we can apply many different tools. No, for instance, so we set up the molecules, the two proteins, and then we do sampling with methods like pyDock or setDock, or we can use also some other stochastic sampling methods like pyDock that we developed also some years ago. And all the models that are generated, they can be in a pool, and they can be scored by this energy function, which can be complemented by machine learning scoring functions like IRA PPA. We can also include the distant restraints from the external data, or SAX data, which is a low resolution structural technique. We can impose symmetry restraints and so on. Okay, so there are many modules that one can use in this pyDock pipeline. So pyDock is being implemented in a server, so it's publicly available. It's been also optimized for high performance computing. It's parallelized, so for optimal use of supercomputers. That was developed actually in the times where the group was at the Barcelona Supercomputing Center, and we are continuing doing improvements now that we are at the SIC. So, as an example of one of the recent developments from a couple of years ago, this is the pyDock DNA web server, which is an evolution of the protein-protein docking approach with that is extended to modeling the interactions between proteins and DNA. Okay, so we optimize some parameters for the specific characteristics of DNA, and then we found that the results, the predictive rates for a benchmark for a set of cases in which we benchmark the tool are actually even better than for protein-protein. No? So except in those cases in which DNA can be very flexible or can be distorted when interacting with the protein. Okay, but for the rest of the cases, even with minor distortions, we are able to find correct solutions in reasonable number of cases. Okay, and this tool is being popular. No, it's being used for many different cases of protein DNA interactions, and even some users are applying this for protein RNA. So even we didn't develop this for protein RNA, and it's not really benchmarked for protein RNA, but it seems that some people found it useful. No, so technically it's possible. I think that we should have like a specific parameters for RNA, but in principle it's possible to use as well. Okay, so the second part is very important because whenever we develop new tools, new methodologies, we need to validate them. We need to test them, and one big opportunity to validate this in blind conditions has been CAPRI. CAPRI is a community-wide experiment in the spirit of CASP that has been going on from more than 20 years ago. So we have participated in all the rounds, and also in the last CASP editions, there has been a session dedicated to multimolecular assemblies that was organized jointly with CAPRI, and we have been also participating in all of them. So basically the setup is that when there is some new complex that is available for prediction, but the structure is not yet available for the community, so it's being held before being released, and so everybody can participate in order to try to predict this model. Okay, basically, like here is an example of our participation in one of the last CAPRI rounds. Usually, in addition to homodimers or heterodimers, there are also highly challenging cases like trimers, hexamers, multimolecular assemblies in which we need to impose symmetry, and also we usually use all kind of information that we may have. And so until very recently, our approach, our strategy was to use different servers for modeling of the interacting subunits, and then we use a parallel run execution of pyDock on one side. So we generated docking models with pyDock, but also we use template base. Okay, and then we in those cases in which template was available, so we use template base, and then we collect all the models and use pyDock energy scoring to rank the final models. So the experience is been very encouraging. So in general terms, we can say that throughout the years, as average, we can submit correct models for around 60% of the cases, which is among the top participants. We have better results in the scorers section. Scorers is just the identification of the models submitted by the rest of the groups. Okay, so in the scorers we don't need to generate models. So in general, we can say that it's been very encouraging, and also it's been forced us and pushing us to develop the new tools for the challenges. No, and here is an example of one very challenging case in one of the last CAPRI editions. That was interaction between a dimeric protein and two molecules of double-stranded DNA. So in that case, we use we had AlphaFold available. We will talk later a little bit about it. We use AlphaFold for just for building the model of the protein, and then we use pyDock to generate the dimeric models of the protein, and also pyDock DNA to predict the interface between protein and DNA. And then we generated thousands of these solutions, these models that were combined in order to have the ones that are not clashing, they have better energies, etc. So at the end of the process, automatically we selected the models that have better energy, and our rank model was acceptable one. We were very happy to see this. Actually, we were the only group that submitted correct models for predictors and scorers in this challenging case. So this is showing the capabilities of pyDock and pyDock DNA, okay, for predicting the structure of complexes. And I think it's very important. It's very interesting to see a general view of the benchmark of protein-protein dockings in general. So very recently, we did some benchmark together with other groups of our tool pyDock and other docking tools in a set of cases, and we found that automatically we obtain success rates between 20-40%. So that would be the state of the art of automatic docking. That means that in 20-40% of the cases, we can have a successful case within the top 10 predictions, okay? Scored by the scoring functions. So if we compare with the performance that the top groups provided in some of the last editions of CAS and CAPRI, that performance was a little bit higher, around 60-70%. But the reason is that if we divide if we classify these cases in the easy and difficult, we understand why this performance. So in the easy cases, like you can see here, these are cases in which they are template-based, and usually here the performance is very large, 90 to 100% of success rate. But on these cases that are difficult, that are defined as difficult, because usually they don't have template-based, so we have to rely on just automatic docking, then the performance, as you can see, is similar to our internal tests. Okay, so that would be the state of the art. On one side, the bottom message is that the state of the art of protein-protein docking is still at this level. And on the other side, if there is in a real case, if you have a possible template, you have to use it. Okay, so it's better to use homology modeling. But everything changed some years ago, 6 years ago, when artificial intelligence was applied for protein structure prediction in a very efficient way, thanks to AlphaFold from DeepMind. In the Cas13, well, this method, automatic method based on artificial intelligence, was better than any other human predictor. And 2 years later, AlphaFold 2, improved version, it was not only the best than any other predictor, but also provide the correct models for virtually all the targets, even very difficult targets. Okay, so now the method is very consistent. Everybody is using it routinely, and actually is providing structures for the majority of individual proteins. And actually, as everybody knows, that's why the creators, together with David Baker, that has been working in the field for many years, they received the Nobel Prize in Chemistry a few months ago. But the question is AlphaFold also useful for protein-protein complex structure prediction? And that question was answered just a couple of years ago. Okay, in the Cas15 was the first time that AlphaFold multimer was available, a special version for protein-protein complexes, and was participating in the predicted in the targets that was proposed for the multimeric targets. And it was clear from the results that the AlphaFold multimer automatic application is better than traditional methods like pyDock. But what we've observed is also that there were some groups that obtained better results than just AlphaFold multimer automatic prediction, and it's because they apply some strategy in which they combine AlphaFold with including uncertainty on the multiple sequence alignments, or or they combine with traditional docking and scoring tools. Okay, so this is one interesting point. Okay, so there is always a way to improve AlphaFold for complexes, and also is some other interesting conclusion is if we see the numbers of the cases that were predicted by the top groups, they were 76%. So not all the targets were predicted by these tools. Okay, so these success rates in these numbers actually was not this number was not much larger than 2 years before when AlphaFold was not available. Okay, so it was better, but not dramatically better. And this was confirmed in the last Capri round that was evaluated a couple of months ago. In that case, well, in our case, for instance, we used pyDock. We wanted to test pyDock in combination with AlphaFold. So we generated many different models with AlphaFold, different versions, including AlphaFold 3, which was available at the time of the experiment. And then we used pyDock in order to help to identify the correct models. Okay, so with this strategy, we had very similar results to AlphaFold 3 automatic predictions, perhaps a little bit higher, a little bit just. But there were other groups that were able to combine these AlphaFold approaches with some other strategies. Okay. Again, if we take a look to the numbers, to the success rates, actually they were very similar to the ones that were obtained 2 years before. It's true that there were different targets. Okay, so the comparison is not completely fair, but somehow it indicates that there are still some targets, some protein complexes that are difficult for the prediction for AlphaFold, even for the AlphaFold 3 version. Okay, so we need to improve somehow to find the ways of improving these predictions. Some of these targets are flexible cases or protein RNA, but also antibody antigen complexes. Okay, so we did a recent benchmark on a set of antibody antigens and trying to use pyDock in order to improve the models generated by AlphaFold 3. So we generated hundreds of models with this tool. But what we found is that in the majority of cases, AlphaFold 3 was not able to provide a high accuracy model. Okay, and on the other side, there were a few cases in which all the models were high accuracy. So that leaves only a few cases, a few complexes in which there were some high accuracy models in which we could try pyDock scoring. In those cases, it's true that pyDock, we observed that it can help to identify the correct cases, the correct models, but I think we need to do more work in general as a community to improve. In those cases in which AlphaFold is not working, we need probably to extend this strategy of AlphaFold, generate more models, or maybe to came up with some other approaches. Okay, so this is current work. But my message will be that pyDock, I think, is also an interesting application. No? So it tried to improve to detect the correct models generated by other tools like AlphaFold. So finally, in the last 5 minutes, I would like to talk also about some applications of pyDock for the energetics of protein-protein interactions. Okay, some years ago, there were the group of Alexander von Wann did some general tests. They wanted to ask the question of can the docking scoring functions are able to predict the binding affinity of protein-protein complexes based on the protein-protein complex structure. So they explored different scoring functions as well as other functions that were specifically developed for energy description. And then they found that pyDock scoring function was one of the best scoring functions for predicting binding affinities. And this is interesting because we never developed this scoring function for predicting binding affinity. So the goal was to identify the correct orientations. So it's true that the overall correlations with experimental data is not great, but what this indicates is that there is some signal in the scoring function of pyDock that we can use to for problems in which energy is important. Okay, and one of these problems was also recently explored. One of the problems is the identification of physiological from non-physiological dimers in crystal structure. Sometimes they are crystal structure in the asymmetric unit in the crystal structure. We can see interactions that can be crystal packing, or sometimes it's difficult to distinguish if they are real or not. Okay, so we had this activity as part of the 3D bioinfo community from Elixir. So together with many other groups, we participated with our tools. No, we explored with a pyDock and also some descriptors that they were implemented in our chart PPI server. If they were able to do some discrimination, so we found that pyDock was able to do some partial discrimination of physiological and non-physiological dimers, but together with the rest of the descriptors of the other groups, the community found a machine learning model that that improved this detection. So this is showing another different application of pyDock, and also another application is the trying to understand the impact of mutations on binding energy. So it is known that pathological mutations very often are involved in protein-protein interactions. So it's very important to localize these mutations and see if they are at the interface of a protein-protein interaction. But not only that, it's also important to describe the energetics, because sometimes two residues at the same interface can have different effects when they are mutated. Okay, so previously, we had collected the database of experimental data on binding affinity changes of more than 7,000 mutations that were affecting protein-protein interactions with all the structural data. And this has been used for us and for other groups to develop more methods to try to predict the impact of mutations. It's been annotated in PDB knowledge base. What we found is a way of calculating the pyDock binding energy atom by atom, and then collecting the contribution of each side chain and this was an estimation of the binding affinity changes of mutation of every residue to alanine. Okay, well, this is a calculation that takes seconds because it's part of our pyDock scoring function. And we obtain similar results to more sophisticated calculations like alanine scanning using long molecular dynamics. We found that there was a correlation with experimental values. So when we apply this to a set of cases from SCEMP2 that were not included in SCEMP1, just to avoid overfitting, we found a correlation between 0.4, 0.5. Okay, it's not great, but it's at the same level as other methods like Foldex, MCSM, very popular methods that are used for this task. And then some of them actually has been optimized using machine learning. Okay, so in our case, we didn't do any optimization. This is just a descriptive work, and we see this correlation. So the method has been implemented in the pyDock NRS web server that can be freely used. And what we observed recently is that if we use AlphaFold models for the complex instead of the crystal structure, we can further improve this correlation with respect to the experimental values, which is kind of curious. Okay, so it seems that for this task, AlphaFold models are better than the crystal structure. So we are now working on extending this approach with two other mutations, with the squirrel. This is one of our line of research. No? So this is also a nice application of pyDock, not docking, but this is just the scoring function, what is being useful. And just to show here some examples of the different cases in which we have applied this methodology in collaboration with experimental groups in order to interpret mutations for which there were structures or identify residues that were relevant for this interaction in that case, but also it can be useful to optimize to protein design and design of interactions. No? So in that case, in collaboration with Jose Casasnovas, we are working on the optimization of nanobodies against some variants of COVID. Applying all these methodologies, we are providing some potential mutations that later are tested in the laboratory. Okay, so just to finish, I thank the members of my group and also all the tools that I have presented here based on pyDock and all these applications are actually the work of many different groups, many different people that has been in my group, and also many collaborators. So I thank you for the attention, and that's it. So I think we have some 10 minutes for questions. You can just open your mic and ask questions. Thank you.